Greetings. My name is Katie Kimberling. I would like to introduce the Capstone project for the Masters of Science in Applied Data Science for Team 8. Our project, entitled Breaking the Lead Optimization Bottleneck in Drug Development for Emerging Infectious and Drug-Resistant Diseases, uses generative artificial intelligence to create new broad-spectrum antiviral analogs to hasten the drug discovery timeline in critical times of need. This project is the brainchild of a wildly and widely talented group of students. Lyndon Conrad Marut brings a background in chemical engineering to the table and was instrumental in the oversight of maintaining chemical relevance in the project. Jordan Torres has a strong background in computer science and machine learning and acted as the project manager for the coding portions. Katie Kimberling, with a master's in biostatistics and epidemiology, helped drive a relevant project focus using data science to quickly combat emerging infectious diseases. Together, we were able to develop a project that tapped into each of our areas of expertise while simultaneously stretching all of us to develop additional skills. The main problem and motivation driving this research is twofold and can be illustrated using the recent global COVID-19 pandemic, but is certainly not limited to coronaviruses. COVID erupted as a viral mutation to which humans had little to no immunity. It was highly transmissible, could be asymptomatic, and cause significant illness and even death. COVID is not the only culprit. Emerging infectious diseases such as Zika and Ebola, or microbes that have mutated resistance to current drug therapies, have established an arms race between the mutation and spread capabilities of infectious diseases and the capabilities of science and chemistry to create medicines to fight them. Viruses can mutate and spread much faster than current scientific methodologies can create treatments and cures. Even when a chemical compound is discovered that successfully kills the intended target, the virus, it then needs to undergo strenuous testing and regulatory oversight to determine human efficacy and safety. This is known as the lead optimization bottleneck, in which many drugs may be hits, they kill the target, but are bunched up in the human trial phase, creating a clogged bottleneck before the drug can pass through to become a viable lead. Our main idea was to use data science to speed up the drug discovery process, saving countless lives and billions of dollars by identifying viable pharmacologic treatments and cures much more quickly using data science, generative AI, and machine learning. The basic premise of our solution is simple, but not necessarily easy. We set out to harness the ability of Gen AI to make exponentially rapid computations in a chemical space. Specifically, we constructed a model that ingests known, usable antivirals and breaks down their text-based molecular structure, known as a SMILES string, into tokens that can be read by a computer. Comparing molecular structures to known leads, Gen AI in our model can then take new analogous molecules that both successfully attack the target and check out as viable for humans by passing through a filter called Lipinski's Rule of 5, which my colleagues will discuss further. The typical chemical structure of a molecule that we all learned in chemistry is depicted on the top left, showing atoms and bonds linked together architecturally in a pictograph. A computer cannot read that and make sense of it. So another way of representing a molecule is that of a SMILES string, as shown on the top right. While this may seem like a random assortment of letters, numbers, and symbols, it is actually a highly regimented way of translating the picture of a molecule into a text string that can be parsed, read, and understood by a computer model. My colleague will go more into the data preparation and pipeline, but initially we extracted the SMILES data for 87 broad-spectrum antivirals for systemic use, meaning these are molecules that have passed through testing and regulation to be approved for human use. We created a filter designed to assess solubility via hydrophobicity and lipophilicity called Lipinski's Rule of 5. We used RDKit in our pipeline. RDKit is a widely used open source toolkit for cheminformatics and machine learning and was leaned upon heavily in this project. Because our data harvested human usable chemical compounds from an open source chemical database, there were no problems with missing data. We decided to use Lipinski's Rule of 5 to assess the ability of a drug hit becoming a lead. Directing your attention to the graph on the far left, we see that although Lipinski-compliant molecules represent a substantial portion of the data set, over 50%, the presence of non-compliant compounds in the data suggests that antiviral agents often extend beyond traditional small molecule design rules and can still retain clinical viability. The middle graph indicates that molecular weight of the antivirals was strongly right skewed, with a few very heavy and large molecules pulling the mean molecular weight to the right of the distribution. The far right graph shows that Lipinski-compliant molecules exhibited a narrow unimodal distribution of molecular weight. That is the flatter, pancake-like picture in the right of the violin plot, while non-compliant molecules displayed extreme bimodal density. This indicates a highly heterogeneous chemical population. Lyndon will now walk you through more of the data pipeline and methodology.
Thank you, Katie. For the data preparation stage, we began by extracting antiviral compounds from the ATC J05 classification within Chemable, which provided a curated set of molecules with known antiviral activity. Each record included SMILE representations along with associated molecular properties and metadata. We then performed several pre-processing steps to ensure data quality and consistency. This included removing salts and incomplete entries, converting all molecules into canonical SMILES format, and validating structural integrity using RDKit. These steps were critical to ensure that only chemically valid molecules were used for downstream modeling. Next, we conducted feature engineering by computing key molecular descriptors such as molecular weight, log P, and hydrogen bond donor and acceptor counts. Using these features, we implemented the Lipinski rule of 5 as a filtering mechanism to assess drug likeness and generate binary indicators for model input and evaluation. Finally, from a governance perspective, we worked exclusively with publicly available chemical data, which minimized privacy concerns. However, we prioritized building a fully reproducible pipeline using Python and RDKit to ensure that our workflow can be reliably extended or validated in future research or laboratory settings. So now going into our overall methodology, including both the data pipeline and model architecture. We started by sourcing antiviral compounds from the ATC J05 class in chemical. We then performed pre-processing steps such as removing duplicates and salts, standardizing molecules into canonical SMILES format, and validating structures using RDKit. Next, we engineered features by calculating molecular descriptors and applying Lipinski's rule of 5 to assess drug likeness. We also tokenized the SMILE strings at the character level, which allowed us to use them as input for our sequence-based model. The dataset was then split into training, validation, and test sets using an 80-10-10 approach. This was because we did not have a large amount of data we were starting with. On the modeling side, we implemented an LSTM-based generative model, which is well suited for learning sequential patterns in SMILE strings. The model generates new candidate molecules, which we then evaluate using RDKit for chemical validity and filter for duplicates. Finally, we assess performance using metrics such as validity, uniqueness, novelty, and Lipinski pass rate, which together give us a sense of both the chemical correctness and drug likeness of generated molecules. Our end-to-end pipeline is designed to take known antiviral molecules and transform them into new potential drug candidates through a structured and multi-step process. We begin with data acquisition, where we pull antiviral compounds from Chemable. These molecules serve as our foundation because they already demonstrate known antiviral activity. Next, in the data preparation stage, we clean and standardize the molecules by removing salts, validating structures, using RDKit, and converting everything into canonical SMILES format. We also engineer key molecular features and apply Lipinski's rule of 5 to assess drug likeness early in the process. Once the data is prepared, we move into modeling, where SMILE strings are tokenized and used to train two different approaches. First, we implemented a naive baseline model, which generates molecules by mutating existing SMILE strings. This gives us a simple benchmark to understand whether more advanced models are actually learning meaningful chemical structure. We then implemented an LSTM-based generative model, which was selected because SMILE strings are sequential representations, and LSTMs are well suited for learning patterns and dependencies in sequence data. This allows the model to better capture the underlying syntax and structure of valid molecules. After comparing the two approaches, the LSTM ultimately performed better in generating more novel and chemically meaningful molecules, even though validity rates still require improvement. Finally, we apply post-processing and filtering, where generated molecules are validated again using RDKit, duplicates are removed, and candidates are filtered based on drug likeness criteria. We then evaluate performance using metrics such as validity, uniqueness, and novelty. Overall, this pipeline demonstrates how generative AI can move from known compounds to new candidate molecules in a scalable and systematic way.
For modeling, we tested two different approaches to generate new antiviral molecules. First, we built a baseline model that randomly mutates existing SMILE strings. This gives us a reference point to understand what happens without any learned structure. Then we implemented a character-level LSTM model, which treats SMILES like a language and learns patterns in the molecule structure. SMILE strings behave similarly to language, where each character represents part of a molecule, so sequence-based models, like LSTMs, are well suited for capturing these patterns. Because of this, the model isn't memorizing molecules, it's learning how valid chemical structures are formed. The goal wasn't just to generate molecules, but generate ones that are both valid and novel. Since this is a generative problem, traditional metrics like accuracy don't apply. Instead, we evaluated performance using domain-specific metrics. We looked at validity, which checks whether molecules are chemically correct using RDKit. We measured novelty to determine whether the model creates new structures rather than reproducing existing ones. We also applied the Lipinski rule of 5 to evaluate drug likeness, which is important for real-world applicability. Finally, we compared similarly known antiviral molecules to ensure the generated outputs remain relevant.
We evaluated both our baseline mutation model and our LSTM generator across training, validation, and test sets using 4 key metrics. These are validity, novelty, Lipinski compliance, and similarity to known molecules. One of the main patterns we see is that the baseline model consistently produces higher validity rates and more unique valid molecules. But this makes sense because it is simply modifying existing SMILES strings, so it stays closer to the known chemical space. However, the LSTM model shows a key advantage. It achieves perfect novelty, meaning all generated molecules are new and not just small variations of existing ones. And this is important for drug discovery, where the goal is to explore new chemical space rather than replicate known compounds. We ultimately chose LSTM as our primary model because of this ability to generate novel structures. While it does have lower validity rates and some instability, especially in the test set, this is a common trade-off in generative models. With more training data and tuning, LSTMs have strong potential to improve both validity and chemical realism, making them more valuable for discovering new antiviral candidates compared to a rule-based baseline.
This artifact is the final layer to our project and acts as a decision support tool. A user starts by inputting a SMILE string, which represents a known molecule. This makes the system flexible, since it can start from any known molecule and explore variations from that starting point. The system then generates a new molecular analog using our trained LSTM model and displays both the original and generated structures side by side. It also compares key chemical properties like molecular weight, log P, hydrogen bonding, and surface area, so users can quickly understand how the molecules changed. We include Lipinski's rule of 5 as a built-in filter to assess drug likeness, along with similarity scoring to show how close the generated molecule is to the original. From a usability standpoint, this makes the model's output interpretable and actionable instead of just raw text. However, it's important to note that this tool does not predict real-world drug efficiency or toxicity. It's meant to support early-stage exploration and prioritization, not replace lab validation. Overall, this turns our pipeline into something that is interactive and something that someone could actually use to explore candidate molecules. Our results showed a clear trade-off between the two approaches we tested. The baseline mutation model produced more valid molecules overall, with a validity rate of about 3.8%. However, these were mostly small variations of existing molecules, so the level of novelty was fairly limited. In contrast, the LSTM model produced fewer valid molecules, around 2.8%, but every valid molecule it generated was completely new. In drug discovery, that distinction matters, because the goal isn't just to reproduce what already exists, but to explore new areas of the chemical space. Another important result is that all of the valid molecules generated by the LSTM pass Lipinski's rule of 5, which suggests stronger drug-like properties. So while the model produces fewer candidates, the one it does generate are more aligned with real-world constraints and more useful for downstream evaluation. The key takeaway here is the trade-off between quantity and quality. The baseline gives you more options, but the LSTM gives you more meaningful options. One major factor influencing these results is data size. We only had 87 antiviral molecules, which limits how well the model can generate and generalize more complex structures. These models together show that generative AI can produce novel and viable candidates, but its effectiveness is closely tied to the amount and diversity of the data that is available. This project focused on addressing a major bottleneck in drug discovery, which is effectively generating and evaluating new antiviral candidates. We developed an end-to-end pipeline that takes known antiviral molecules, represents them as SMILES, and then generates new analogs using AI and filters them based on chemical validity and drug-like properties. To summarize our work, I'll break this into two parts: what we built and what we found. From a project outcome perspective, we built a competitive generative AI pipeline for antiviral molecule generation. The LSTM model was able to learn structural patterns in smiles and generate novel molecule candidates rather than simply reproducing existing ones. We also incorporated RDKit and Lipinski's rule of 5 to filter and evaluate candidates in a way that reflects real-world drug constraints. In terms of key findings, one of the most important results was the trade-off between validity and novelty. Simpler methods produced more valid molecules, while LSTM produced fewer but more meaningful and novel candidates. Importantly, the valid molecules that were generated by the model consistently met drug likeness constraints, which strengthens their practical relevance. This also highlights the potential of the pipeline to expand chemical search space beyond known compounds, which is a key challenge in drug discovery. Finally, we translated this into a web-based artifact, creating an interactive tool that allows users to explore and compare generated molecules in a more interpretable way. While these results are promising, there are still important limitations and areas for improvement, which leads directly into our next steps.
One of the biggest limitations was the small dataset size with only 87 molecules. This limited the model's ability to generalize and learn more complex chemical patterns, which also contributed to lower validity rates compared to our baseline. We also saw that LSTM had difficulty capturing long-range dependency in SMILE sequences, which are important for representing more complex molecular structures. In addition, our evaluation was fairly limited. We primarily relied on Lipinski's rule of 5, which is useful for drug likeness but does not fully capture toxicity, biological activity, or how molecules actually interact with targets. Because of these limitations, several next steps are critical. First, expanding the dataset is likely the most impactful improvement. By incorporating additional drug classes like antibiotics or cancer drugs, we can give the model more diverse examples and help it learn more complex structural patterns. Second, exploring more advanced models like transformers or VAEs could improve performance. Since these models are better at capturing long-range dependencies and complex relationships in sequence data compared to LSTMs. We also want to improve evaluation by incorporating ADMET and toxicity prediction, which would make the pipeline more realistic and aligned with actual drug development workflows. Finally, improving generation reliability and enhancing our web-based artifact will make the system more usable in a real-world lab setting, where researchers need fast, interpretable, and actionable results. Overall, these next steps focus on making the pipeline more accurate, more scalable, and more applicable to real drug discovery.